Welcome to another lecture. This lecture is about the photosynthetic dark reactions, which are also known as the Calvin cycle or the C3 cycle. And then later on, we will contrast that with the Hatch-Slack pathway or C4 pathway and the crassulacean acid or CAM pathway. Okay, so let's orient ourselves here. For all these reactions, including the ones that we discussed last time when we talked about the light reactions, we are here in the chloroplast. So the light reactions occurred during the day along the thylakoid membrane. We had the photosystems 2 and the photosystems 1 with the chlorophyll that were capturing the photons and were creating these this chain of reactions, of redox reactions. And in the end, what we had was NADPH and ATP, as well as oxygen. So those were our three products: oxygen, NADPH, and ATP. And the ATP is now important because that's going to be used for the Calvin cycle, or the Calvin-Benson cycle, as it's also known, as well as the NADPH. So those products from the light reactions are now going to be used in the dark reactions, or the carbon reactions, in the stroma of the chloroplast. And you will see that that will then generate NADP plus and ADP again, which during the day can then be used again in the cycle of the light-driven reactions to regenerate the ATP and NADPH. And at the same time, what we are doing with these products from the light reaction is we are going to use the hydrogens from the NADPH to reduce the carbon dioxide that comes in from the outside. So basically, we're putting hydrogens on carbons, and that's what this is signifying up here. And of course, those are carbohydrates, sugars. So all of these will be reduction reactions, adding hydrogens, in this case, to carbon. Okay, so here's an overview. And this overview of the Calvin cycle shows you that there are three major parts of this cycle: the carboxylation part, the reduction part, and the regeneration part. And then at some point in this reaction, in this Calvin cycle, glyceraldehyde 3-phosphate, an intermediary of this pathway, is sort of siphoned off from the Calvin cycle and is then used for starch or sucrose formation. So this is the point where the carbohydrates are taken out of the cycle and then produced into sugar and then either stored or used at other times. You will see that these figures from your book are color-coded. So whenever you see the blue part or the green part, that has to do with carboxylation or reduction or regeneration or the carbon output. So look for these colors, and then maybe that helps you a little bit. And the other thing that I want to point out is this designation of carbon numbers. So how many carbons in a chain are we talking about? So there's a lot of names later on, and you'll see that maybe that quickly becomes overwhelming. But if you just keep track of how many carbons are in a chain or in a molecule that we are looking at a time, then that makes it easier to sort of keep track of the carbons. So carbon dioxide here is a C1 chain, not much of a chain really, it's just CO2 with one carbon. But ribulose 1,5-bisphosphate, which is the acceptor of the carbon, that would be a C5 compound. So C has 5 carbons, 1 carbon. At this point, it splits into 2 times 3 carbons. 1 3-carbon compound is used for the siphoning off into the sugars, and 1 C3 compound is used in the regeneration of the C5 acceptor. And you see, of course, that C3 and C5, that doesn't add up. So there are multiple copies of these that go into making multiple copies of those. And just some of the glyceraldehyde 3-phosphate is siphoned off into the carbon output. We'll look at the details in a second. So what I just said is that we have 3 important steps in the Calvin cycle: carboxylation, reduction, and regeneration. And I also said earlier that we used the ATP and the NADPH from the light reactions, and then we're using carbon dioxide here from the atmosphere to produce the glyceraldehyde 3-phosphate, which then becomes sugar. Here's the formula for the glyceraldehyde 3-phosphate. But your book and the upcoming figures also show you these chemical formulas. All right, let's start with the carboxylation reaction. So that's the first step in the Calvin cycle. So ribulose 1,5-bisphosphate is right here. And when we talk about all these names and these compounds with long, chemical names, I don't really expect that you remember all of them. What I do hope that you will get out of this is that you can look at a compound name and you can sort of see what it must mean. So, put the parts together and sort of get an idea of what the compound would look like sort of in general. So if you look at ribulose 1,5-bisphosphate, you should be looking for 2 phosphates, and they should be at the 1 and the 5 carbon. And ribulose is a sugar. OSE always indicates that it's a sugar. And so ribulose 1,5-bisphosphate, would have 5 carbons and 2 phosphates at the 1 and 5 position. And so you see that here, you see that here. And likewise, if you look at the 3-phosphoglycerate, so if you're looking at the parts of the name that you recognize, there's a 3 and a phospho, so that at the carbon position 3, there should be a phosphate. And then glycerate, or glycerol, that has 3 carbons and 3 hydroxyl groups. ATE, 8, is always the ending for a carboxylic acid that is decarboxylated. So in other words, an organic acid that has given off its hydrogen. And so you see that up here, 2-oxy-COOH, that is our carboxylic acid. And when that is off, then it's negative. And so then that would be the glycerate. Otherwise, it would be glyceric acid. But glycer should ring a bell that it's 3 carbons. The 3-phosphor position is right here. So that's kind of how I hope that you look at names and sort of get an idea of what the compound looks like in general. Okay, and so in order to get this stoichiometrically correct, there's a bunch of numbers here that are added so that you can get out of ribulose bisphosphate a phosphoglycerate. And so what will happen here is that 3 carbon dioxides are used, waters, and the acceptor molecule. So that's 3 times 5, that's 15 carbons. 3 times 1 is 3 carbons, so that's 18 carbons going in, and coming out is 18 carbons with this 3 carbon chain times 6, so that's 18. So 3-phosphoglycerate is the compound that is the result of the carboxylation reaction, the fixation of carbon. This is the important step where carbon gets taken out from the atmosphere and essentially made into an organic substance. So the next part of the Calvin cycle, the reduction part, then uses a lot of ATP. So you see that the ATP is used up and phosphates are added. Now we have a phosphate group at the 1 and the 3 position. So now we have a 1,3-bisphosphoglycerate. And then we come to the reduction reaction. So here we have the NADPH. So hydrogens are added to this substance here. And remember that adding hydrogens in biochemistry essentially is a reduction reaction. So you're adding the hydrogens from the NADPH, and you are getting glyceraldehyde 3-phosphate. So there's an aldehyde group, there's a phosphate group, glycer, 3 carbons. And then of these 6 glyceraldehyde 3-phosphates, 5 of them are used as the regeneration of the acceptor. So all of those go back to produce a 1,5-ribulose bisphosphate, and only 1/6 of these of this glyceraldehyde 3-phosphate pool is then siphoned off as the output to become fructose, glucose, or starch. Yeah, and then there's the regeneration reactions. And the regeneration reactions in the Calvin cycle are quite complicated and are really not just a linear pathway that will lead you back from the glyceraldehyde 3-phosphate back to the acceptor molecule. Your book has all the reactions in it. You see that there's a number of various different compounds, intermediates that are made, and they fuse and they break and they fuse again, and in the end you get the acceptor. So if you're interested in looking at each of the individual steps, you should look at the book and see them all. Important for you to remember is that there's a carboxylation reaction, a reduction reaction, and then a regeneration phase of the pathway. All right, so that's really the Calvin cycle, or Calvin-Benson cycle, as it's also known. The enzyme that is part of this step, the ribulose bisphosphate carboxylase oxygenase, that's the name of the enzyme. That whole thing is the name of the enzyme, also abbreviated as Rubisco. That enzyme, as you see in the name, doesn't only have carboxylation activity, so adding a carbon dioxide, but it also has an unfortunate side reaction that it can also catalyze, and that is the oxygenation reaction. So adding oxygen to ribulose bisphosphate is not useful. It's simply not useful. There's nothing good about it. And it's a relic from the past, long time ago, when there was much, much less oxygen in the atmosphere. So plants were evolving, they were using carbon dioxide, and there was hardly any oxygen in the atmosphere. Plants were really the ones who added the oxygen into the atmosphere as we have it now. So the evolution of that enzyme wasn't under any pressure at that time to create a enzymatic reaction that would suppress the oxygenation part of it, because there wasn't any oxygen around. If you think about this evolutionarily, given the fact that that enzyme, which is the most abundant enzyme on Earth, right? So if you took all the enzymes and measured them, that is the most abundant enzyme on Earth. Apparently, plants are fine even though there's this oxygenation reaction that occurs. So there must have been payoffs or trade-offs that allowed the plant to change its pathways such that it can deal with this oxygenation reaction rather than coming up with a new enzyme strategy for fixing carbon dioxide. And that is the content of what I will tell you about now, which is also known as the C2 pathway. So the C2 pathway, or photorespiration pathway, is a pathway, it's a salvage pathway. It doesn't do the plant any good except that it fixes an error. It fixes this error of having oxygen incorporated erroneously into the ribulose bisphosphate. So I write here in red, Rubisco has affinity for both oxygen and carbon dioxide. This results in photorespiration. So it's called photorespiration because it is coupled to photosynthesis, but it's really a respiration reaction where you are using oxygen instead of carbon dioxide. So the C2 cycle is a salvage cycle. It just basically cleans up after this enzyme when it picks up oxygen instead of carbon dioxide by error, so just by coincidence. And this happens all the time, right? It's not just a rare event. It happens all the time to a certain percentage of all reactions. The more oxygen in the atmosphere, the more likely this will happen, because that much more likely the enzyme will grab the wrong molecule. So it's quite a complicated pathway, and it's a cycle, but it's not just a circular linear pathway. So all of these reactions are part of the C2 cycle. So what do I want you to learn about this? I don't want you to learn all about the individual parts. I don't want you to learn all the names of the enzymes and the substances that are made in between, but I want you to understand what is generally happening. And so I put these numbers on here, parts of the pathway, and I'll point out what happens at these particular parts. And then on the next slide, I have what I'm about to tell you written out so that you can copy that from that slide if you want for your notes, or use the slide as your notes. Okay, so this is in your book. You can probably only see the names if you blow it up on your slides. But this is where it starts. So we're in the chloroplast here, and this is the first part of the reaction. where ribulose 1,5-bisphosphate, that's the compound here, picks up oxygen. This is the oxygen. So it picks up oxygen instead of carbon dioxide. And the first thing that happens in the salvage reaction is that the 5-carbon group breaks up into a 2-carbon and to a 3-carbon group. So the 5 carbons become a 3-carbon and a 2-carbon. And the 3-carbon group is phosphoglycerate. So 3-phosphoglycerate, that's a useful substance, right? That is the substance that we get from the incorporation reaction in the Calvin cycle. So that is useful. That can just go straight back into the Calvin cycle. This is the Calvin cycle up here. So 3 of the 5 carbons are useful and can go back and can be used. But this part here, the 2 carbons, that is 2-phosphoglycolate. So glycol, that's a 2-carbon alcohol. Glycolate is the carboxylic acid anion. So we have the 2-phospho with a phosphate group at one of the carbons. That is our substance that is the problem of this oxygenation reaction. So 2-phosphoglycolate is toxic, so it has to be detoxified, it has to be gotten rid of. And the way that this pathway does it is that it not only gets rid of it by completely degrading it, but it actually makes it into a useful compound that costs a little bit, but in the end, a useful compound is made that you will see back up here, that is 3-phosphoglycerate, the same compound that you have here, but it has 3 carbons. There's only 2 carbons here. So somehow a carbon has to be added here, and then that can also go into the Calvin cycle, and then everything is fine, and the problem is repaired. But what happens in the way to detoxify glycolate, the 2-phosphoglycolate, to produce a 3-phosphoglycerate? That's the gist of the C2 cycle. So let's take a look. So oxygen was incorporated, 2-phosphoglycolate was created, and the glycolate is now exported out of the chloroplast into a body here that's called the peroxisome. So the peroxisome in plants is a place where fatty acid metabolism occurs. So I wrote that back here because that's probably an organelle that you may not have spent much time thinking about. So fatty acid metabolism occurs here, but in addition to that, there are also enzymes around that will help the plant detoxify the 2-phosphoglycolate. So the glycolate, after dephosphorylation, gets added into this, transported into the peroxisome, and it produces, these enzymes produce out of it glycine, so that's an amino acid. And then this is the important step. So 3A is where 2 glycines, with the help of water and NAD+, fuse essentially and produce serine. So this now has 3 carbons, and 1 carbon is split off. So 4 carbons here, and then we have the 1 carbon splitting off here, and the other fusing so that we have 3 carbons here. In the end, we want 3 carbons, right? So now here we already have regenerated a 3-carbon compound, and that 3-carbon compound now has to be reduced again. So there's a reduction reaction here. There's a phosphorylation step, so that costs ATP. And so serine is then shuttled back through the peroxisome into the chloroplast, and essentially at that point, when it's made back into a 3-phosphoglycerate, added back into the Calvin cycle. So what's the problem? Well, the problem is that you are losing 1 carbon that was previously incorporated into sugars, and that had a cost, right? It costs ATP in the Calvin cycle to incorporate carbons. And if you then just split it off again and have to pay ATP in the end as well, that's a cost to the plant that is not useful. That's energy loss. So that's the problem of the photorespiration cycle. Of course, the upshot is that the plant has detoxified its glycolate and can go on without any other problems. So what I told you just is all written up here, so you can take a look at that again. What are the products? What are the numbers of carbon dioxides? Salvaging this, what is the cost? and then in the end, you're feeding the 3-phosphoglycerate back into the carbon cycle. So these numbers go with the numbers here in the picture. And I'm pointing out here that we have the 2 times 2 carbons, right? So that's the glycines into 1 time a 3 carbon. That's the serine with this carbon coming off. So from experience, I know that students find this confusing, to say the least. This is a confusing pathway. So I try to put this together into a different view. It's the same thing that I'm showing you on this next slide here, on this slide. Same exact thing, but just sort of attacked in a different way. So let me try to walk you through this, and maybe you find this more intuitive than the other figure with all the little chemical names in it. So what I have here is I split this into 2 parts, the oxygenation reaction or the carboxylation reaction. And I have a cycle here, sort of a cycle, and here as well. And I'm focusing on the number of carbons in the compound. And then I'm pointing out where the carbon loss occurs and where the energy loss essentially because of that occurs. So let's walk through this quickly, and then you can look at this more closely and see if this makes more sense to you than the previous slide. So after regeneration of the acceptor, we have the ribulose bisphosphate. In order to make this stoichiometrically possible, we have to work with 2 C5 compounds, so ribulose bisphosphate, 2 molecules of that. Right, so 2 of those, that's a total number of carbons of 10. If we now add oxygen, we're adding no carbons, right? So we're still at 10. And then I said that this substance is breaking apart into 2 carbons and into 2 carbon chains, one with C3 body. So we have 6 carbons in 2 C3 bodies. That's the 3-phosphoglycerate. So that comes from here. And then the other one, the glycolate, only has the 4 carbons. And so that is the remainder, right? So out of the 2 times 5, we now have 1 times 6, 1 times 4. And that again is split into 2 molecules and this into 2 molecules. So out of these 2, these fuse, right? The 2 times C2 bodies, they fuse. They make the serine here. That's a C3 body. That then eventually becomes 3-phosphoglycerate. But that only now has 3 carbons, and this is the 1 carbon that gets lost. So 1 of 10 carbons is lost here, and I'm saying this is 10% of the original input into this reaction that's lost. And then this carbon chain is added to this pool, so these 2 come from here, this one comes from here. That gives you a total number of 9, and that goes then back into the regeneration part. And then that can go back into hopefully a carboxylation reaction in the next step. But you see that you lost a carbon. And so what I'm writing here in green is that 1 round of this oxygenation loses 10% of previously fixed carbon to carbon dioxide. Right? So that's what comes up here. So that's a problem. There's no gain. There's only loss in the oxygenation part. If you look at the carboxylation part, this is basically just the Calvin cycle again. We have the 2 molecules of the ribulose bisphosphate, 10 carbons total. We're adding now carbon, right? So this is the incorporation of carbon. Now we have 12. So that's a total of 4 times C3 bodies, right? So this is a C3 body, 4 times of those, the 3-phosphoglycerate. That can all go back either into sugars or into regeneration. And it's not just splitting 3 to 1. That would be straightforward, but that's not exactly how it happens. So the stoichiometry would actually be even more complicated with a lot more molecules overall to make this work. I just wrote in here how the split generally is. It's about 2 carbons that go into sugars and 10 carbons go into regeneration. Of course, there's no 2 carbon compound that's splitting off here, right? We talked about that. That is a 3-PGA that becomes sugars. But this is just to sort of count carbons to see what is used and what is used for sugars and what is going into regeneration. So that's why I wrote here in green, 1 round of carboxylation incorporates 17% of the carbons into sugars, that's this, and moves 83 of the carbons into acceptor molecules. So that's this. 83% goes back here, 17% goes in here. Nothing is lost, there's only gain here, right? So here you have no you have no gain, you have only loss. Here you have 17% of the carbons that are gained that are then moved into sugars. So that's our C2 pathway. And now we have the C4 pathway that I want to talk to you about. So the C4 pathway, also known as the Hatch-Slack pathway, those were the guys who first understood most about it. That pathway is important because it is an adaptation to climates, a certain climate adaptation. I'll tell you a little bit more about that in a second. But what I wanted to start this out with is by pointing out the slides that you saw in Lab 1, where you looked specifically at C3 and C4 plants. And what I pointed out there was that C4 plants have an adaptation that C3 plants don't have to their vascular bundles. So these are the vascular bundles. And here's the xylem, here's the phloem, xylem, phloem, xylem, phloem. And then here we have this ring of cells that are surrounding the vascular bundle. And so these are the bundle sheath cells, also known as Kranz morphology. Kranz in German means wreath. So that's sort of a wreath around these the vascular bundles. So a C4 plant, that is characteristic for a C4 plant. And plants that are C4 plants, the best known is corn, maize. So that has a C4 metabolism. And C3 plants don't have any of that, right? So here's a vascular bundle, here's a vascular bundle, no bundle sheath cell around, lots of holes here, or spaces, you should say, where in the spongy mesophyll there's gas exchange going on. You don't really see that here. So why is that important? I point that out in a second when we talk about the metabolism. So on here, I just wrote what I just told you, so you can read that up again. Small bundle sheath cells, photosynthesis done equally in all parenchyma cells. Here we have the Kranz anatomy and the bundle sheath cells that are surrounding the bundles. Okay, so it will become clear, hopefully, why that is good for the plant in certain environmental conditions. So let's first take a look and see what happens in this pathway. So atmospheric carbon dioxide is fixed to an acceptor, and that acceptor is phosphoenopyruvate, or PEP, phosphoenopyruvate. Right? There's a phosphate, enol, so there's the En, that's a double bond, En. Chemical chemists will know that anything ending in En has double bonds. And then ol is a oxygen, so has both a double bond and the oxygen, the alcohol group, and that alcohol group is phosphorylated. So with the help of PEP carboxylase, that's an enzyme, right? Phosphoenopyruvate carboxylase, that's the enzyme. It incorporates carbon dioxide from the atmosphere into the first stable compound, being oxaloacetate. You know oxalic acid, if you remember, maybe from the TCA cycle. So that's part of the TCA cycle, as is malic acid or malate. The interesting thing here is not so much who the acceptor and the first stable compounds are, but that carbon dioxide is not added as carbon dioxide, but with the help of carbonic anhydrase, carbon dioxide and water produce hydrocarbonic acid, so H2CO3. And we talked about that with respect to pH in the soil. You may remember that from one of the earlier lectures. And the anhydrase pushes the equilibrium to this anionic form, so the hydrocarbonate form. And that is the form that can then be incorporated by the PEP carboxylase into the oxaloacetate. And I'm circling the carbon dioxide here in red so that you can follow that carbon that is incorporated. So that's the carbon here, that's the carbon here now, right? It's getting added to this 3-carbon chain, now it's a 4-carbon chain. Now it gets reduced, right? There's an addition of a hydrogen, so it gets reduced to malate. Here's our carbon still. And then these reactions that occur in the mesophyll cells, so in the outward facing cells towards the edge of the leaf, that is then shuttled through a specific transporter, the malate transporter, into the bundle sheath cells. So these are the inward facing bundle sheath cells. So these are close to the vascular bundles. And here we have malic enzyme using NADPH and splitting off the carbon dioxide. And that carbon dioxide then goes into the Calvin cycle. And that's the whole C3 cycle. That's the same as the Calvin-Benson cycle that we discussed earlier, the C3 cycle. So at that point, we have what's left over of the malate, which is pyruvate, and then pyruvate gets shuttled back into the mesophyll cells. With the use of ATP, it becomes phosphorylated, and we have the phosphoenolpyruvate restored as the acceptor of the next molecule of carbon dioxide via carbonic anhydrase. So this extra pathway here, this circular pathway, is placed in front of the Calvin cycle, of the C3 cycle. It's just an additional cycle. C4 does not replace C3. C4 is added to C3. And you see that it actually costs ATP. Now, why is that useful? Why is that of any help to the plant? We'll talk about that in a second. So here are a few notes of stuff that I just said. This is a preface to the Calvin cycle. What it does is it enriches the tissue with carbon dioxide. This tissue is enriched with carbon dioxide. And oxygen is removed because it's not actively removed, but since it's further away and it's sort of separate from the outside where the oxygen is, you have an enrichment of carbon dioxide over oxygen, right? So it's not scrubbing oxygen away, but it's adding more carbon dioxide. So the ratio becomes higher, and therefore the likelihood that the Rubisco enzyme will use carbon dioxide instead of oxygen in its first reaction step. And so that will reduce overall photorespiration. Okay, so here are all the steps that I mentioned earlier. You can read them. You can look at the the predicament that there's photorespiration happening and that the C4 cycle is preventing that. Here are all the individual steps that I pointed out. Let me also say that another predicament or adaptation is that in warm climates, higher temperatures decrease Rubisco's carboxylase activity. So both of these enzymatic activities are temperature sensitive. And so warmer temperatures decrease the carboxylation activity. And so therefore, enriching carbon dioxide with this pathway is preventing the additional photorespiration that otherwise would occur in an enzyme that is negatively affected by these warm climates. So that is why plants that are growing in warm areas have adapted the C4 cycle, while plants in the C3, in colder climates, are usually just using the C3 by itself. No C4 that's sort of put in front of it. And the reason for that is that it costs, right? It costs a little bit of ATP, but the cost of the C4 cycle is lower than the cost of photorespiration would be. And so therefore, it's better for the plants. It's advantageous to have the C4, C3 pathways than to deal with photorespiration. All right, and now the last pathway of what I want to talk about today is the CAM pathway, or the crassulacean acid metabolism. So Crassulaceae, that is a family of plants. I'll show you some pictures of what Crassulaceae look like. Essentially, they have thick leaves. They are sort of, thick, fleshy leaves. I'll show you a few pictures in a second. That pathway is really quite similar to the C4 pathway, except that it's, people say it's temporarily separating the incorporation of carbon dioxide and the Calvin cycle, while the Hatch-Slack pathway doesn't do this temporarily, meaning over time, but spatially, over space, right? So they have two different cell types where carbon fixation originally occurs and where the Calvin cycle is, while in the CAM pathway, this is separated by time, where we have night and day reactions. So why does this happen this way? Well, and how does it happen? So at dark, at night, and when it's dark, the stomata are opened. And that means that when it's hot during the day, plants would lose a lot of water through open stomata, right? So they close the stomata during the day, but when they close the stomata during the day to prevent water loss, they can also not pick up any carbon dioxide. So if they pick up carbon dioxide at night, they can prevent the loss of water. But if you pick up carbon dioxide at night, you may not have enough ATP around, because ATP comes from the light reactions, right, which require sun. So what the CAM pathway essentially does is it takes carbon dioxide, it stores it, it sequesters it in the vacuole, and then during the day, when ATP is around, but no carbon dioxide because the stomata are closed, then it remetabolizes that carbon dioxide back out of the vacuole and then puts it into the Calvin cycle. So let me point out the individual steps. Again, I put numbers on the important parts, and you can find the the steps that occur here in the next slide for you to read or review. So what happens in the first step? We have carbonic anhydrase, same enzyme as in the Hatch-Slack pathway. Carbon dioxide from here or from the mitochondria, so respiratory carbon dioxide can be used. The carbonic anhydrase makes hydrocarbonate, just like in the C4 pathway, and PEP carboxylase, just like in the C4 pathway, takes this from phosphoenolpyruvate, same adaptation as in the C4 pathway, and makes oxaloacetate from that, right? Also the same as in the C4 pathway. And then we have the same types of steps, except that malate is now not shuttled into a different cell, but it's shuttled into a different cell compartment. And that compartment is the vacuole. So now we have malic acid in here, not malate, so that's not the protonated form, but the acid form. And when the light comes on, then the enzymes change their activity and the PEP carboxylase becomes inactive. And at that point, malic acid is transported out into the cytosol again as malate, and then transported into the chloroplast, right? This is the chloroplast here. And then carbon dioxide is split off. That's the same as it happened in the bundle sheath cells, right? We had malate there. We had carbon dioxide being split off in pyruvate mate, and then the carbon dioxide is shuttled into the Calvin cycle, and the Calvin cycle then just produces sugars and starch, just like we talked about in the C3 pathway. That's the same exact pathway. Right? This is the C3 pathway. So the CAM pathway, just like the C4 pathway, is a pathway that is in addition to the C3 pathway, that's added on in front of the C3 pathway. Examples of plants that use this are cacti, some orchids, and then crassula. So crassula, that's the Crassulaceae, right? That's one of the plants of the family that gave it its name, the Crassulaceae. And in the crassula, there are plants that you might know, sedum. Those grow here as well. They're small, they have yellow flowers, they have fleshy leaves. And this pygmy-weed jade plant, you might have that in your dorm room on the windowsill, and it's very hard to kill that plant. They have these, there's about yay big. Where am I? Yay big, and then thick leaves. If you forget to water that, no big deal. And pineapple is also one of them. So you can take a look at these, and I want to point out that the advantages here are that water is conserved up to 10-fold over a C3 plant. So that is substantial, right? That is an adaptation for dry climates, not necessarily for hot climates, but for dry climates. In some species, CAM can be activated or deactivated depending on water availability. And so overall, we have the disadvantages and the advantages, right? Advantages are water conservation, but the disadvantages, of course, are that it's not quite as effective as having the two reactions of photosynthesis, the light reaction and the dark reaction, going at the same time. So taking the carbon dioxide and storing it away and then remetabolizing it later costs a little bit and is less efficient. So we have overall reduced growth. We have less photosynthesis. We have relative slow growth and relatively small plants as a result of that. So those are CAM plants. And here's an example of all these CAM plants. Here's a jade plant, and then here a number of other Crassulaceae plants that have the CAM pathway. All right, and that is it for today. See you in class.